Okay, so skeletal muscle contraction. So most of us know that we can contract and relax a muscle, but what is going on physiologically there that is enabling that skeletal muscle to contract? So we know that there are three types of muscle tissue in the body. We have smooth muscle and cardiac muscle, that is involuntary. And then we have the skeletal muscle, and that is going to be voluntary. We can will our muscles into contraction. So what's going to happen is that we're going to have an action potential traveling down a motor neuron to the neuromuscular junction. That's going to be the union between the synaptic end bulb of the motor neuron and the sarcolemma, or the cell membrane of the skeletal muscle. When neurotransmitter, the specific neurotransmitter for a motor neuron is going to be acetylcholine, is released onto the sarcolemma, that will depolarize the membrane of the skeletal muscle, and that action potential will travel down the sarcolemma, dive down into structures that are called T-tubules, and those T-tubules are going to have voltage-gated channels on them that will be able to release calcium from the sarcoplasmic reticulum, which is the endoplasmic reticulum in the muscle cell. But the sarcoplasmic reticulum is a repository for calcium ions. And so calcium is going to play an essential component in muscular contraction. So now let's take a look at the contractile unit of skeletal muscle, which is the sarcomere. So the lateral boundaries of the sarcomere are called Z-discs. And so when we have one Z-disc and another Z-disc, those are forming the lateral boundaries of the sarcomere. Skeletal muscle is also going to be striated. So when you look at it under the microscope, you can see that there are alternating light and dark bands. Those bands have specific names. So where we just have the thin actin filaments, so these are the thin actin filaments, and no thick myosin filaments, so these are the thick myosin filaments, where we just have those thin filaments present, that's going to be considered the I band. So you can remember that there is the letter I in the word light, and so the I band is going to be light histologically. The A band is what we can see here. That's where we're going to have the overlap between the thin actin filament and the thick myosin filaments. And you can remember that there is the letter A in the word dark. So this A band is going to look dark histologically. And so we're going to have alternating light and dark bands. Some other components of the sarcomere include the H zone. So that's the space between the ends of the two actin filaments. And those dimensions are going to increase and decrease in size with regards to the H zone, depending on whether or not the muscle is contracting or relaxing. Right down the middle is an imaginary structure called the M line. That's marking the middle of the sarcomere. So I mentioned that calcium is going to be important for initiating contraction, specifically in the sarcomere. So once that calcium has been released from the sarcoplasmic reticulum, it's going to make its way into the internal components of the sarcomere here. So where we have the thick myosin filaments, we're going to have these myosin heads and myosin tails. And it's going to be the interaction between the myosin and the actin through a power stroke that will cause the sarcomere to contract. But we have a conundrum here in that there are going to be affiliated proteins here with the thin actin filaments, one being tropomyosin. And upon tropomyosin, we have another protein called troponin in purple here. And when calcium is not present, this protein complex is going to be blocking the myosin binding site on actin. So those cross bridges are not going to be able to form. and we can't get the power stroke needed to bring the Z disc closer together for muscular contraction. But when calcium is present in the sarcomere, when it's been released from the sarcoplasmic reticulum, it can bind to troponin. When calcium binds to troponin, that causes a conformational change in the protein tropomyosin, and so it will move away from the myosin binding site on actin. And so once that has occurred, we can have the myosin heads binding to the thin actin filaments, and we can start a power stroke, which will culminate in muscular contraction. So that's an overview of the sarcomere and how action potentials are sent down motor neurons to the neuromuscular junction that excites the sarcolemma. That action potential then travels down the sarcolemma, dives down into the T-tubules, opens up voltage-gated calcium channels so that we can have calcium doing its thing at the sarcomere to enable cross-bridge formation to occur.